So thank you very much, and thank you for the invitation. It's really nice to be here and to present our work today. So today I will speak about single-cell approaches to collective behavior. The main interest in our lab is to understand how individual cells are first regulated, and then how bottom-up these individual cells can create multicellular systems that have higher order properties that are really necessary to create then organs and tissues. This multicellular system comes from the coordination of many individual cells. This means that cells, individual cells, need to take individual decisions, but they need to be coordinated at the tissue scale. That means they need to know where and when they are. So, and for this coordination at the different biological scales, we need to have methods first for individual cells to transmit information forward, and there are many different ways cells have been doing this, from different gradients or morphogens or different waves that can have different scales. But then we need to have a feedback from the higher level scale, from this multicellular system, from the tissue, back to individual cells to then control the individual cell fate. And these are often also mechanics and different extracellular matrix. So in order to do this, what is important is that genetically identical cells, individual cells, are able to form this emergent tissue level property. But this big gap in between the individual cells and this emergent tissue level properties are population of cells. And so this population of cells have a very intrinsic emergent property that is variability at the population level. A population of cell is intrinsically variable, and this depends on both intrinsic state and extrinsic cues that individual cells feel. So different metabolic cues, different adhesion between the cells, or different, for example, cell cycle state. And these individual cells then have different probability to differentiate. And this is what we call symmetry breaking. So an individual cell here will differentiate and have an irreversible change that then drive these emergent level properties. So our main question is how individual cells sense the extrinsic cues from the environment to take individual but coordinated decision at the tissue scale. And again, just to reiterate, this is a problem about biological scales. So we need both spatial and temporal methods to be able to measure both very fast event and that might be at the plasma membrane of an individual cell that have consequence days later at the tissue scale. So we have been using single-cell RNA-seq to identify different, for example, cell types. In this case, for example, you see that individual cell type are very easily characterized and seen in single-cell RNA-seq. We can then infer trajectories from this data. However, we have a very big problem that this is what we call decision-making across biological scales. So in this population of cells where actually decisions are making, this is where there's the majority of the variability between the cells. And when we start looking at these cells, this is what we can define cellular states. These are very different to define by single-cell RNA-seq, because as also mentioned, these are dependent on extracellular cues, extracellular matrix, and cell-cell contact. Moreover, it's not only mRNA, but protein and protein stability and localization that is very important, and the phenotypic cellular state of the individual cells. Moreover, this variability that we see at the population level is not an intrinsic variability or a random variability, but these are metastable cellular states in their environment that then can have, that is a very dynamical and reversible type of movement between this metastable cellular state. And the last point I want to make is that stemness and this decision-making is a property of a population of cell. It's not an individual cell that will do it all alone. It's always a cell embedded in its tissue. And so to address this, in the past years, we have been developing different methods to approach these problems. And on one side, we use hundreds of thousands individual organoids with high dimensional measurement and a very big perturbation space, and the dynamics using light sheet imaging and tracking of individual cell. Some of you might know some of the work we have been doing in the lab on intestinal organoids, but today I really want to show you some new data that we have that are unpublished. And so again, a lot of our work really focus on the variability and the heterogeneity in population of cells and on symmetry breaking. And if we want to go at the first really symmetry breaking event during development is the formation of an anterior-posterior axis. And this happens when you have between the E5.5 and E6.5, the formation of, here at the bottom, there are these cells that will start migrating up here, and this is gastrulation. So the migration of these epiblast cells and the formation of the mesoderm and the definitive endoderm. This first step is very tissue intrinsic. And this has been shown because if you take these cells in vitro, you can break symmetry. So, and this is like an overview of many different systems that have been derived, but in many of them, symmetry breaking is embryo intrinsic and is not dependent on extracellular cues. Sorry. And the extraembryonic tissue really provides the niches and the directionality. We have been using one system that was developed in Alfonso Martinez Arias' lab, and this is an EB-derived gastruloid that then has the 3 germ layer. So just to give you an idea of how the system works, these are 300 mouse ES cells that then grow up to 5 to 6 days. Here, there are more than 500 micrometer. They have symmetry breaking, and this happens also with the activation and a step of Wnt activation between 48 and 72 hours. There are the formation of the 3 major axes, there's gastrulation, and the expression of many cell types. There are clearly some limitation, but this can be done both with mouse, human, and zebrafish embryos. So just to have a look at how this works, we have these 300 cells between 48 and 72 hours. We have the upregulation of brachyury that then this asymmetric distribution of brachyury really define the first anterior-posterior axis. And you have this axial elongation. So this is clearly, you have something happening before, then this Wnt activation and this elongation axis. As I said, one of our main way of approaching the problem is to start also with a very broad perturbation spectrum. So what we did, we design an high content imaging based gastruloid screen where we add compounds at 3 different time points. The first one is adding the compound before the Wnt activation and during the Wnt activation and during the elongation. This is the library of 84 compounds. It is a full automated setup that then also uses 40 gastruloid per condition. We do the segmentation of all the different organoids that are stained by DAPI, icadirin, brachyury, and SOX1 that then stain for the 3 main germ layers. Then we segment both the full gastruloid, and then we also create, here as you see, 100 segments to have really an anterior-posterior type of analysis. Then we take these gastruloid features and we cluster them to find what we call a phenotypic space and a phenotypic landscape of gastruloid development. That looks like this. Here you see there are like 9 main phenotypes that cover the different space. And here in this UMAP, you see that every dot is not an individual cell, but an individual gastruloid that include all the features of the different ones. Here you see that you have the control and color-coded all the different features. But one aspect that was very interesting is that we have the 3 different time points. So can we then dissect the 3 different time points at which we add the compounds? So here you see is a UMAP where we add the compounds before, during, and after the Wnt activation. And I won't go in detail of all the data, but here there was something that you can see is really striking. This purple phenotype, that is this icadirin, that phenotype with a little bit of SOX1, is present only if we perturb before the Wnt activation. And indeed, there are all these compounds that have a very specific phenotype only when perturbed at the first time point. And then at the later time point, they just become phenotype 5. So this is what we were mainly interested. So our question at that point was, what happens before Wnt activation? Most of the field really thought that this Wnt activation would drive symmetry breaking and the asymmetric expression of Wnt for brachyury. But really, what we tried at that point is to set up a time course experiment of gastroloid development. So the time course, we take the gastroloid, exactly the same protocol, but then we fix them at different time points. We can infer a trajectory, and again, this is not a RNA-seq trajectory where the different dots are, the different points are single cells, but these are gastroloid at a different time point. So we can infer an image-based trajectory of gastroloid development. And then we can start looking, and here you see the expression of brachyury, and we can have both at the whole gastroloid level or the different segments over time, in this case is over pseudo time of this trajectory. And what we find was quite striking in the sense that at 24-hour, you see this SOX2 is expressed everywhere. But at 42 hours, we have this core of SOX2 cells, and the outside are SOX2 negative. So this is very spatially organized. And here you see the quantification of 100 or 1,000 gastroloid overlaid. But that's what was more, here you see also nodal, but what was most impressive is that when we activate with Wnt, brachyury expression is only in the negative cells. So in the outside of SOX2. So we thought of different ways. Again, is it the SOX2 expression? But what seems to be happening is that at very early time point, here you see there's a fibronectin core that stays for also later on. So you have this extracellular component of fibronectin that create a niche that probably maintains this SOX2 core. So what we think is that we have a very early extracellular matrix core of fibronectin that create this niche that maintains this SOX2 core. When then we that create the first asymmetries. Then the Wnt activation just reinforced this, making only the SOX2 negative reacting to brachyury and then driving the full formation of gastroloid. That means that we have a Wnt response that on the periphery drive the primitive streak. But the core, we really don't know what's happening. Then that we decided to do a single cell RNA-seq time course. And in here, we did similar to the imaging, the time course. Here you see 24, 36, 48. Then there's the Wnt activation. We jump this gap, and then we have the 60, 72 hours, and 84. Here, what you see, every dot in this case is a single cell that comes from population of gastroloid. Something that was quite interesting that at a later time point, we have this population of cells. So here you see 60, 72, 84. But in this early population, you have a mix of all the 3 time points. When we start looking at SOX2, you see that this core of early population, like if they're stuck in the later time point after KIR activation, express SOX2. But in early time point, we don't have a clear separation or a clear cluster of SOX2. We see like a normal progression. When then we color code for brachyury, this is clearly expressed in the other cells that are not the core cells. When we color code different cell types that have been characterized in the early mouse embryo and have been published, here you see that most of the cell type happens in the brachyury expressing cells that are later on. That then brings me back to the early time point I made. These are all cellular state. We really cannot find a clear cell type or cell state or a cell cluster. We tried 2 million ways of clustering. So when we start looking at these population, this one that it looks really that is stuck, we wanted to know what happens to this population that react differently to the Wnt. And to our big surprise, what we found is very early pluripotent marker. So this is ZFP42 or KLF2. These are very early pluripotent marker. And when we indeed also stain for NANOG, we see that at 60, we still maintain this pluripotent core. And when we color code the signature genes, we see that this very early time point, that is these cells that are stuck at the late time point, But again, in this early time point, we don't see a very clear progression. We then try to take the early time point to see if, looking more at these ones, we could find any patterns there. And what we see is that doing also a velocity analysis, there's a main single trajectory through these data. That there's not an actual bifurcation. It's not that the two population of cells go into different direction. Then we did, here you can see some of the clustering and some of the analysis of the velocity. And then we created a score. This is the score that is the overlap between the cells and the score of cells that maintains pluripotent. And what is quite clear that you see that there's, when you go through latent space and score, you have these cells that have a very high score that are very early in latent space. Because in a way, what latent space and all this trajectory do, they're creating a molecular progression, and where then we can map time. But really what we are looking at is a single molecular trajectory where some cells here at 48 hours are still stuck. And indeed, when also we analyze 48 hours, you have these very early cells that in latent time are still very early and have a very high score. So what we think at this point is that the formation of gastroloid has a very interesting process where we have the score of fibronectin that makes sure that the core has a much slower progression. So these are not differentiating in different, these are exiting pluripotency at different speeds. And the extracellular matrix and fibronectin makes this progressing much slower. The outside, probably driven both by nodal, making the cells more competent for Wnt activation, then are the one reacting to Wnt and then driving the formation of the primitive streak. And so we have a system where, and what we find very interesting is that even if you have a continuum of progression, so from slow to faster, there's a clear threshold probably of SOX2 and ratio with some of other components where some of the cells then move forward and the other one go back to pluripotency. So there's no intermediate response. And with this, I won't go in some of the validation that we also have been doing on some of the signaling that drive this very early process that we found on the screen, but I hope I could convince you that combining different technology that are not only spatial and like with single-cell RNA-seq, but also combine a full space of perturbation, because I think perturbation space and being very deep in your phenotyping during the perturbation is extremely important. And this is then helping us to get much closer to decision-making across biological scales and to understand how cells can coordinate across these biological scales, transmitting information forward, but also having a feedback to then try to coordinate at all the different scales. With this, I'm at my last slide, and I would really like to thank especially Marietta Zinner that started a full new system in the lab and the only one not working on intestinal organoid, Simon Suppinger, and Ilya Lukonin for all the part on the gastroloid. Here, all the lab, you can see it as an intestinal organoid with also Paneth cells in the crypt. Our collaborators, Mattia Lutov and Fabien Theys, all our funding, the FMI and all the facility, and all of you for your attention.
Thank you, Prisca, for an absolutely stunning talk. There's a couple of questions already popping up, so I'll ask them to you. So D Who asks, to what extent do you think this fibronectin niche is applicable to in vivo embryogenesis?
So the role of fibronectin has been very well characterized much later, but there are some papers that clearly show some fibronectin role very early on. And what we think, this is what we don't know if there is a local fibronectin. We don't know if the cells that respond to fibronectin are actually the one making the fibronectin. And I think would be very interesting to look if there is something between cells and how they coordinate that. Some cells make it, might be a certain layer. But there are some now report that also that fibronectin seems to be very important in maintaining a pluripotent state. So it might be a local niche that they are generating for themselves.
Yeah, I feel like those things are so difficult to test because we have very little control over these, like changing the ECM and local niches and things, right? I mean,
yes. So we are trying to. So there are ways where we can then change and engineer the system, also with optogenetics, and then changing maybe the expression or flooding the system with the fibronectin. So we are trying a bit to play with these niches because both there and also in the intestinal organoid, the self-generated niches, I think, are getting more and more important.
Okay, I've got a question here from John Murray. Sorry if I missed it, but is the different rate of progression for internal versus external cells driven by the same gene expression program occurring at different paces, or by different genes being expressed in each group of cells, or a mix in the analysis you did?
So we have been looking if there would be something that would create that there's a different gene expression program, but they look that they go through the same differentiation. So we tried to add then Wnt at different times and different size to then control the timing. And it seems just that it's really a single progression and they're just going slower in that, so that the program is the same, is exiting pluripotency.
That's pretty wild.
But it's just that the speed is different. Then what determined the speed? It's and what determined the response? Because the thing is that the exiting pluripotency seems to be a continuous, but the response is binary. You go forward and you go backward. You don't stay. So I think that's quite interesting.
That's pretty cool. So Carmen Gerlach asks, what is it that the ECM core does to slow down differentiation? Shield cells in the core from external differentiation factors? Question mark.
Yeah, so I think that this is a very interesting question, and I think it's a combination. So part of it might be that the ECM with some different signaling, so it could be FGF, would be maintaining a bit the core, and we see that part of our perturbation is, for example, FGF that then keeps the fibronectin all the way. But on the other side, you have nodal that is really outside. And I think this is also quite interesting because this is a full 3D system. But if you know the system that Brinvalou and Wormflesh developed, that is the 2D patterns, you see that nodal has this ring also outside. So probably this ring that is outside in the 3D and in the ring in 2D makes the cells faster competent to a Wnt response. So I think it's a combination of the core of extracellular matrix that probably via FGF maintains, like is a 2I type of state for pluripotent cells. And while the outside, thanks also to nodal, makes it more competent for the Wnt.
Cool. I'm going to ask one more question. So I'm vested in this question too. So fantastic talk, Dagan Segal says. Technical question, how do you get throughput of imaging of thousands of whole embryos?
These are gastroloid. Yeah, you should ask Marietta. It's she's finishing her fifth year of PhD. She took, because many of these gastroloid were taken by hand, one by one, and re-put it in plates. So she redesigned the full assay, and I think that was almost a year to redesign it in special plates. So she's also looking for the special plate where then she has one single gastroloid per well, and then many wells for every condition. So this is for the embryo, we are not there yet, clearly from that's more complicated, but the automation of the culturing is very important, and especially maintaining batches of medium, batches of matrigel, batches of anything you are considering, because also for the reproducibility is very important. Yeah. In this system, these ES-based systems, is very hard. We see that there are problems when even you freeze the cells before. In the passage before where they're frozen, so you need to make sure that they're frozen correctly to then be able to do all the steps. So it's the culturing and the automation there is quite complicated.
Well, the answer often to how you do awesome stuff is very painfully and carefully.
And yes, and a bit control freak on some aspects, especially on the culturing of the cells. I think that's quite important, and I think also analyzing broadly, because I think that the problem with these systems where you risk to then analyze just a subset of your structures, then just create artifact, and then you're focused on certain problem, and then when you have some certain perturbation, you're looking again at a subset while being very broad and looking at the full population. I think it's very important.
Yeah. Thank you so much.